Good morning, everybody. So welcome to this meeting. This is a meeting of the NIHR Statistics Group, and we are the Laboratory Studies section of that group. So my name is Alice Stitch, and I head that group. Just to give you a little bit of background about the NIHR Statistics Group. So the purpose of that group is actually to bring together statistical methodology and the application of that. So that's what we're trying to do in this session. So we're thinking about qPCR, and we're splitting that out into the sort of laboratory side of things and then the statistical application. So as part of the NIHR Statistics Group, we're trying to bring people together. So we're trying to link up with the statisticians, and we're really trying to sort of bring together, particularly for this group, the laboratory sciences side of things and statisticians, so we can learn from each other. There are several working groups as part of the NIHR Statistics Group. So you'll see those on the side there. We're laboratory studies, but there are others if you are interested in getting involved in any of those. So today's event, so I'm delighted that we've got our speakers today. So we've got Ben Towler and Chris Jones, and they are both representing Brighton and Sussex Medical School. So it's excellent to have them here with us today. And the way the session will work is we have got the theme of qPCR. And so we're going to think of this from the laboratory side and then from the statistics side with some questions in between. If you could please provide all of your questions through the chat function, and we will look at those during the sessions and at the point of the scheduled question times. You may have seen a box pop up as well, telling you that the session will be recorded. So just to let you know that that's happening. Future meetings of this group, we do have a couple of meetings planned for early next year. So one is on basic statistics training. If anybody does have any comments on what you would like to see there, please do send that through to me in the chat function or separately via email. And we have another session that we're putting together at the moment, thinking about repositories of data. We have a mailing list, so don't worry about writing this down. I'll put this in the chat function for all of you. So if you would like to join that, that would be great. I'll make sure that's available to you as the session goes on. So that's just my very quick introduction to this session, and I'll hand over to our speakers for you now.
So I'll share Ben's talk.
Hi, so today Chris and I are going to talk to you about the design and analysis of quantitative PCR experiments for lab scientists and statisticians. So because of the diversity of people that we're expecting here today, I'm going to go right back to basics. To start off with, you've got the session overview. So we're going to talk about introduction to qPCR and its uses. We're going to talk about qPCR chemistry, different chemistries and how it's used for quantification. We're going to go through all the important steps of an experimental setup, the selection of reference genes, crucial controls that need to be done. And then I'm going to finish by talking about calculation of relative expression and using delta-delta CTs and calculating fold difference. And then Chris is going to take on from me and talk about analysis considerations, how to graphically represent your data, and then important statistical considerations for the design of your experiments. And all of these have very, very common pitfalls. So we're going to try and help you avoid those. So like I said, because we have a vast diversity of expertise here today, I'm going to go back to the basics, so to the central dogma of biology. In our nucleus of our cells, we have our DNA. Every time the cell divides, it's replicated, so it's copied by an enzyme called DNA polymerase. Our DNA encodes genes. The functional product of that is quite often proteins, but to get to the protein, we need to get it out of the nucleus. And so this occurs in a process called transcription. DNA is transcribed into what is called messenger RNA or mRNA. These mRNAs encode for proteins, and these are produced from the mRNA in a process known as translation. And obviously, talking about PCR, what we're talking about here is the replication of DNA by an enzyme known as DNA polymerase. So this is the enzyme that is involved in the PCR, so the polymerase chain reaction, which we'll talk about all day today. So a basic polymerase chain reaction, so this is not qPCR, is it uses DNA polymerase to amplify specific regions of DNA that you want to amplify. So it could be a specific gene of interest that you want to make more of. This is used in a number of settings. So it's used molecular cloning, it's used in diagnostics, for example. So the way it works is 3 steps in what is called a cycle. And so we have our region of DNA of interest here. So our DNA is double-stranded. We have a 5' to 3', and we also have the reverse strand, and these are complementary to each other, meaning that these bases, A pairs with T and C with G. And so what happens here is we then have primers. So these are specific small sequences, so in general around 20 nucleotides, which bind to a complementary region in the DNA. So this would be a sequence complementary to your gene of interest that you want to amplify, for example. Okay, and we have a forward one and a reverse one. So one that's complementary to the forward strand and one that's complementary to the reverse strand. We also have what's called dNTP. So these are the nucleotides. So these are the individual building blocks of your DNA. So your A, your G, your C, or your T. And we also have our DNA polymerase. At the moment, all these bases are paired up, so they're not able to bind to anything. So what we need to do is we need to break those bonds. And we do that in a step called denaturation, which involves heating, typically to around 95 degrees. And what this does is it breaks the bonds in between the 2 strands of DNA, which then opens the strand and produces something which your primers are then able to anneal to. So I said here we have one complementary to the forward strand and one complementary to the reverse strand. And this is achieved in a process called annealing, typically between 55 and 60 degrees, but it can vary depending on your primers. And so what happens in this step is our primers find the complementary sequence, and this, as the name suggests, primes the DNA polymerase in a process known as extension, where it extends along from the primer and fills in the complementary base to the strand that it's copying. This is a singular cycle, and so what happens after the first cycle is then we repeat this over and over again. So for example, if we were in the second cycle, we then have the denaturation step. So now we're denaturing here, so we now have 4 strands of DNA instead of the 2. And so then during this step, exactly the same thing happens. We have 8 strands after this. The third cycle happens again. So now we have 8 strands, which are then the primers are able to bind to, et cetera, et cetera. And so obviously this process is exponential. So every time we're doubling the amount of DNA material that we have. This is a really efficient way of increasing the amount of material that we have to a level that we're able to use in the lab. And so obviously after the nth cycle, we have 2 to the n copies of our PCR product. And so typically what then happens is we run this on a gel, and we should see a nice strong band at the side. So when we run a gel electrophoresis, the smaller the product, the faster it runs through the gel. So here's an example. So we've got our 4 samples. We tried to amplify this bit of DNA, which is around 500 nucleotides in size. And we can see here a nice strong band. And so this is a typical use of a polymerase chain reaction. But obviously today we're talking about qPCR, so quantitative real-time PCR. And this is a quantitative application of that polymerase chain reaction we've just spoken about. So it's applicable to both DNA and also RNA, which needs to be reverse transcribed into DNA to perform this, but we'll talk about that later on. So there are a number of uses for quantitative real-time PCR. So for example, gene expression analysis, which is what I use it for, and is probably the most common use. So how much of a specific RNA transcript is present in my test samples compared to control. And for this, you can look at mRNAs, long non-coding RNAs, for example, but you can also use specific conditions to look at small RNAs, such as microRNAs. You can also use qPCR for SNP analysis, so single nucleotide polymorphisms. You can use it for copy numbers, and more recently, the COVID PCR test is an example of this. And so how does qPCR differ? So like I said, it's a quantitative measure of PCR. So how does this actually work? The quantitative element is basically provided by the fluorescent dyes and a detector in your thermal cycler or your PCR machine. Without the qPCR bit, we have our PCR, we run it on a gel, and this is what we see. So we see it at the end. So say we do 35 cycles. At the end, we see this band, but we have no idea here, because we've taken it to saturation, we have no idea whether there's more in 2 than 1, for example. So what happens with the qPCR is we trace the amount of DNA material or PCR product over time. And this is achieved by using a fluorescent dye. So at the end of every cycle, the amount of fluorescence is detected. And then as you can see over time, the fluorescence starts to increase. We get this lovely exponential phase here. And then we plateau when we get to saturation. And so what we're seeing on a gel is actually this region up here. So we have no idea if there's more or less in this situation, but you can use, but quantitative PCR will allow us to look much, much earlier on that we wouldn't normally be able to see. There's two major chemistries for this, which I'll go through. There's probe-based chemistry, such as TACMAN chemistry, or there's cyber-based chemistry, and talk about the differences between these. But the choice of chemistry is really crucial for subsequent experimental planning and the controls that you have to do. So personally, I use probe-based chemistry. This is my favorite type of qPCR, and it uses a specific probe in addition to the forward and reverse primers. So previously, I showed you during the PCR, you have the primers, which are complementary to either the forward or the reverse strand. And so that's shown here. But with the probe-based chemistry, you have an additional bit of DNA, and this is a probe. And so what is on this probe? So this probe itself is also complementary to the sequence of interest, so your gene of interest, and that lies in between the forward and reverse primers. It has a fluorophore, but it also importantly has a quencher. So when these are in close proximity, i.e. the probe is intact, the quencher inhibits the fluorescence from the fluorophore. Okay, so under these conditions, we don't see any fluorescence at all. However, obviously during denaturation, we then pull the strands apart. We then allow our forward and our reverse primers to bind. We then have our probe binding in between the two during annealing. And then during the extension step, what happens is the polymerase has 5' exo activity, and this means that it cleaves nucleotides in the way, basically. And so what will happen is as it extends through the probe, we get cleavage of this, meaning that now the fluorophore is released from the quencher and we're able to detect signal. And so every cycle, this happens. And then the camera, like I said, the camera in the machine will then detect how much fluorescence there is. And then obviously over time, the number of cycles will increase. And so this then increases the amount of DNA or the PCR product that is present, which means the amount of probe that binds, there's more of that, which means there's more fluorescence released as the polymerase extends through. And so this then means that over time, we're able to measure the increase in fluorescence. And the good thing about this is obviously here you have 3 different bits of DNA, which are all complementary to your target. So you're much less likely to have off-target binding, which also means you're much less likely to have non-specific results. So cyber is generally held as a cheaper sort of type of chemistry. And this, unlike the probe-based chemistry, doesn't have the probe, it just has your forward and reverse primers. So all cyber does is it binds to double-stranded DNA. So whenever you have double-stranded DNA present in your sample, you have cyber binding, and then that will release a signal. So cyber only fluoresces when it's bound to double-stranded DNA. And so obviously here, what happens again, we have our annealing with our forward and reverse. We then have some double-stranded DNA that our fluorophore binds to. And then as it extends, obviously there's more double-stranded DNA, so there's more availability for the fluorophore to bind. And then again, as the PCR progresses, you're getting an increased number of PCR products, which then again, in turn means that we get more cyber fluorescence, which means that the machine detects more fluorescence. So these are 2 different methods. So just a quick summary of these. So obviously the probe-based is considered more expensive because you have to buy specific primers, whereas cyber is cheaper. It's just a dye that you add into your master mix, or a lot of master mix these days have it already in there. But the advantages of probe, you don't need much optimization at all. So the efficiency and the specificity is pretty much guaranteed by whoever you buy the probe from. Whereas with cyber, you need extensive optimization. So you design your own primers, you then have to make sure they're specific. You have to make sure the concentrations are correct. So for every single cyber reaction that you use a new set of primer pairs with, you really should optimize your primer concentration and test the efficiency and test the specificity, which you don't have to do with a probe-based chemistry. Again, the specificity, the probe-based gives you more specificity purely because you've got 3 regions of complementary DNA, whereas with cyber you only have the 2. So the chances of off-target effects, i.e. binding 2 primers binding somewhere that they shouldn't near to each other, is higher when you've got 2 than 3. Again, because of this, cyber is more likely to have false positives. Obviously, you can get around this with certain controls and making sure your primers are correct. And one of the ways you do that is with a bell curve. So you do it, and we'll talk about that later on. So you have to do a melt curve with cyber to make sure what you're detecting in the machine is a single product, whereas you don't do that, you can't do that with probe-based chemistry. Okay, so these are the two sort of main chemistries that are used. So it's completely up to you which one you use. So now we're going to sort of talk about the important features of a successful qPCR workflow. So I like to think of it as sort of a melting pot of your sample preparation, your RNA, so the material that you're using. So here we're going to talk about looking at RNA expression, so different genes of interest, and the reverse transcription. So this is the process of making your RNA into DNA so we can perform PCR on it. And every single one of these is really crucial that it's optimized and that you have the best quality sort of steps as you can to produce a good qPCR. And so, for example, with the RNA extraction, it's really important that you have a high enough yield, the integrity, so this means how intact the RNA is, and how pure the RNA is, so to make sure you haven't got any contaminants. Obviously, the sample preparation itself is down to your experimental conditions, and you should have the correct controls, etc., based on your experimental condition. The reverse transcription is also really important, so the efficiency and the quality of this. So a good qPCR is heavily dependent on the reverse transcription step. And then again, going into qPCR, we've spoken briefly about the different chemistries and the primers, and obviously within this, the efficiency and the specificity have to be very, very, very good. And then you also have your reference genes, which we'll talk about, which are really crucial parts of the qPCR experiment, and your controls. So these are your negative controls, your positive controls, and also your experimental controls. Okay, so it's really important to try and think of all of these steps. And so if you optimize and you make these as good and efficient as you can, then you're going to produce a good qPCR experiment. So if we go through each of these, so obviously I've mentioned that your sample preparation is down to you, but the RNA extraction itself, so obviously most RNA extractions will involve a standard phenol chloroform step, and mainly lots of people use kits these days for this. But what's important is that you have sufficient yield. Ideally, you'd like to get a microgram at least, but obviously you can do this on a much lower amount of RNA, and it obviously depends on your experimental condition to how much material you can produce. But you have to make sure you can get enough to detect reliably what you want to detect. So you can measure yields in a number of ways. So nanodrop quantification is sort of the most common way of doing this, as long as you have a reasonable yield of RNA. So if you have very, very high yields or very, very low yields, the nanodrop can quite often be sort of random number generation down at that level in my experience. But in general, it's pretty good. The important thing to note here is DNA contamination will be included in a nanodrop quantification. So all the nanodrop looks at is excitation at 260, any nucleic acid basically. So if you haven't performed a DNA digest in your RNA extraction, you should, because it will help you much later on. But you'd have to take into account that if you've got DNA in your sample, then that will affect your quantification by nanodrop. An alternative method of quantification is to use a qubit. So this uses intercalating dyes instead of just a A260 rating on the nanodrop, and will not pick up DNA. So this is another method if it's really important for you to make sure you have the best or the most accurate quantification, which is really important when we talk about determining reference genes, etc. later on. I also mentioned the purity, and so this can be assessed on a nanodrop, again using the 260-280. So this would tell you if you have sort of protein contaminants, or 260-230, and this would tell you if you have salt contaminants, or we quite often get carry through of the organic plates. And ideally, these should be above 1.8, which means your RNA is a good purity. And finally, I mentioned integrity, and so this is how intact your RNA is. So ideally, what an RNA integrity number or a RIN of greater than 7. And so to get a RIN, you have to use a machine known as the bioanalyzer or a tape station. And so what these do is it basically runs a tiny little microfluidic and looks for the presence of the 28S and the 18S ribosomal RNA, and then looks at the ratio between the two. So what you get is a trace here where you have the 28S and the 18S, and you see these two nice spikes and nothing else. And this would tell you that your RNA is very good in integrity, so it's nice and intact. What you'd see is sort of these would sort of collapse and you see a big mound if your RNA wasn't intact. So by all means, don't do this every single time you do an RNA extraction. But when you're optimizing kits, when you're optimizing conditions, it's really important to make sure the extraction methods that you're using are clean and that they are producing intact RNA, and you're avoiding RNase contamination, etc., etc. So if you don't have access to a bioanalyzer, you can obviously also run integrity by running on a gel. And when you do this, for example, you should see two nice strong bands for the 28S and the 18S ribosomal RNA. So this would be an example of some decent RNA. However, an example of some degraded RNA here where we don't see these strong bands, and this will be RNA you don't want to take forward. So this is all part of your optimizing your RNA extraction. So once we've decided we have a good RNA extraction, RNA is really high quality, we have enough of it, it's pure, we need to go into the step called reverse transcription. And this is the production of DNA from RNA, so complementary DNA. So this would be called cDNA. And this is performed by an enzyme known as reverse transcriptase. And so this is making, like I say, our RNA transcripts within the cytoplasm that we've isolated during RNA extraction into DNA that we can then perform PCR. And like I mentioned earlier, qPCR really does rely on an excellent reverse transcription reaction, and you want the efficiency to be as consistent as possible. There are loads of kits available for this, so test a few. There's a choice of oligo-DT or random prime. So the difference here being that oligo-DT will only reverse transcribe polyadenylated RNAs. So these are, for example, your mRNAs and your long non-coding RNAs. If you're interested in other RNAs which don't have that poly-A tail, which is a protective structure of the 3' end of the transcript, then use random primers, which tend to be random hexamers, so random 6 nucleotides. So that will reverse transcribe everything within your RNA sample. So it's also important that you don't use more than the recommended amount. In my experience, if you try and flood it, then there's not enough of the reverse transcriptase enzyme, or for example, and so the efficiency drops, which means you get more variation in your qPCR. So it is important to kind of abide by the manufacturer's suggestions here. Another step is to perform no RT controls. And so these are effectively samples where instead of adding the reverse transcriptase enzyme, you add water. And these are to detect for DNA contamination. So once we're happy with our RT, we have a really high quality cDNA, we're happy, it's nice and efficient. What we need to do is then go into obviously a successful qPCR. And what's important here is a consistent input amount. So obviously the template is crucial because all other reagents are in excess. What you're controlling is the amount of template that goes into the reaction. For example, the polymerase, the primers, the dye, or the probe are all in excess. So there's more of it than you could ever need. And this is to make sure that the template is the limiting factor. This is why it's important to have your quantification accurate as well. So personally, I always put the same amount of RNA into the RT and then the same amount of RT into the qPCR. And this allows me to control it every single step so I can see if there's been a problem. And so if there's no difference, we should expect exactly the same value, right? Which we'll talk about later. So the next thing is obviously primer specificity. So this is crucial. So primer should only bind to your gene of interest. So you should only get a singular product when you do the PCR. And so I would always say if you're going to do, this is normally guaranteed with a probe-based chemistry. However, if you're using a cyber-based chemistry, you should always run your product, test your primers first, run them on a gel, make sure the product is the size you expect it to be, and that it's the only product there. So as I say, this is enhanced by using probe-based chemistry. However, if you use cyber, like I said, you must perform a melt curve. And so this is effectively looking to see how many PCR products you have. And so what you should see is this singular peak. And so this is effectively the melting of your PCR product. So the pulling apart of your double-stranded DNA. And what we see here is a nice singular peak, meaning that your primers are really nice and specific. However, if you have non-specific primers, what you'll see is these sort of more lumpy nature. And this is telling us we have probably 3 different products there. And obviously what we're looking at in the qPCR is a single trace. So what we'd be seeing is a combination of this, which really isn't okay. So we need to make sure that you have nice singular peaks on your melt curve. And then if you've got this, then happy days. The final thing that's really important in terms of your primers is the amplification efficiency of them. So these should be close to 100%. And so by 100%, what we mean here is that it doubles each time. Now, realistically, that's not going to be what's happened every single time. So what you want to have is a range between sort of 90 and 110% efficiency. You can look up ways to do this, and there's plenty of information online. You effectively perform a standard curve, and then you calculate the line here, and what you should have here is a pretty much a straight line. So every cycle, like I mentioned before, every cycle it doubles. And so what you should have is a close to doubling every cycle. And then from there, you calculate your amplification efficiency. And finally, it should be reproducible, obviously, like any scientific experiment. And what's also important is to use the correct controls to enable you to assess false positives and false negatives, but predominantly false positives, because these are a real problem in qPCR. Okay, so the first thing is your very standard negative controls. So, for example, in no template control, you do exactly the same thing in a normal PCR. So this would involve the addition of water instead of cDNA to the mix. And then what this would control for is any false amplification, any contamination, often with your primers, for example. So if you contaminated your primers with cDNA in the past, then you'd get a signal which you obviously don't want, so you'd have to go back and make new primers. But what we shouldn't see any amplification in. I'll talk about it again in the future. Sometimes you see late amplification, but we can use different things to determine whether it's real or not. And the other thing is the no reverse transcriptase controls, which is what I mentioned earlier. So reverse transcriptase is excluded from the reverse transcription reaction, which means that if we get a signal, it's purely because we have DNA contamination, or you might have other contamination, but the most likely effect is you have DNA contamination, or your genomic DNA. So what's important with this is to design, is to design primers across exon-exon boundaries. Obviously, humans, we have massive introns, and so what we try and do is a forward and reverse primer. So these are two exons and an intron here that span the boundary. So realistically, because of the size of introns in humans, we would never get a product here if we had genomic DNA contamination. So this would avoid that problem. However, some transcripts are monoexonic, which means we have to design, or that might, for some reason, we might not be able to design across an intron boundary. And so if your primers bind in a single exon, then it's really important to do these no reverse transcriptase controls to make sure that we're not just picking up genomic DNA. We also need technical replicates. And so this is each combination of primer pairs and cDNA sample. And so this should be done in at least triplicate. And this is the average, and then the average of 3 is then taken. So these are identical. So the way I work is I make a master mix enough for 3 reactions. And then I put 10 microliters into each well of the final qPCR, which then should be identical. And so this allows you to control for pipetting errors and mixing of the right mixes and everything. And finally, I mentioned previously no reverse transcriptase. You can also do reverse transcription replicates. And I'd suggest to do this when you first test your samples. So when you first test the reverse transcription kit, I'd really strongly recommend to do this, because effectively it's producing exactly the same. You put the same amount of RNA into 2 separate reverse transcription reactions, and then the CT should be exactly the same. So your level of expression should be exactly the same. And this is telling you that the efficiency of your reverse transcription reaction is very consistent between samples. And as I say, it's useful to test new RT kits because qPCR relies on efficient and effective RT. Okay, so finally, in terms of controls in general, is the selection of reference genes. And so reference genes you would have heard lots about, and it's really crucial to control for the amount of cDNA put into the qPCR. So all of this so far is based on the quantification by either nanodrop, qubit, bioanalyzer, whatever you use. But importantly, these reference genes should be unaffected by treatment and conditions. So they should be consistent. So I'll talk about cycle threshold in a minute. But effectively, what we're looking at here is our control samples and our treatment samples, the level of expression is very, very consistent. And this is really crucial. And that's because calculation of differential expression is completely dependent upon a suitable reference gene. And we should expect to see this because we know we put the same amount of starting material into the reaction. And the thing is, for each experimental question, you really do need to empirically determine your housekeeper. So typical ones tend to be GAPDH or HPRT1, but it's really important to actually make sure they're correct for your experimental condition. And so because this paper used them is really not an acceptable reason to use it. So, I saw this paper in this that used completely different cells or completely different patients that use this, and so I'm going to use that. That's not okay. And there's so many examples of that in the literature, which have really muddied the qPCR literature. But there's a number of methods you can use. Obviously, use a literature search by all means, but select multiple candidates to test for yourself under your conditions. If someone's done something similar, then by all means use those as a starting ground, but you have to test them yourself. You can use things like GNorm or Norm Finder, which are algorithms. You do the qPCR on your potential reference genes, and then you use these algorithms and they tell you which one is the most suitable. If your qPCR is following a sequencing, which is a lot of examples these days, then obviously select 3 or 4 different references which don't change in your sequencing data and see if they're reliable to use in your qPCR. There's also more statistical models, so equivalence tests are starting to be used, but I don't have any experience in using those as yet. So how do we determine the suitable reference genes? So we do a qPCR, so it's really important to make sure you do this on your samples themselves. They don't necessarily have to be exactly the same samples, but they have to be the same condition and treatment. And so what we do here, so for example, in this hypothetical experiment, we've taken 6 different genes, and then we've run a qPCR on our control and our reference. And what we see here, these are all groups. And so what we see is we see a lot of these are quite consistent. We see some variation here, but in general, we've put the same amount of RNA into our RT, we've put the same amount of cDNA into our qPCR, so they should be very, very consistent. And so from this, we'd then select reference B. So here, reference B is the most consistent, meaning that this is the reference gene we want to take forward and use. We can also do serial dilutions to make sure they respond as they should. But yeah, from here, we take reference B forward. I'd also advise don't use a reference gene which is too low in expression, because then you get natural variation, which is then going to throw your experiment even more. So I've been talking a lot about cycle thresholds. So what actually is the cycle threshold? So the cycle threshold is the number of cycles it takes for the normalized fluorescence value to cross a threshold value. So what we see here is this is our amplification cycle by cycle, the amount of fluorescence we see, then we enter this exponential phase, and then we hit the saturation up here. And so what the software will look to do is it will look to set a threshold at the point at which your reaction becomes exponential. And so here, for example, it's set to 0.216141, for example. And then what the software will then do is tell you how many cycles it took for your trace to then cross that line. And that is your cycle threshold that you then take forward and use. And I mentioned normalized signal. So just to give a quick heads up, so you see here, this is delta Rn and not fluorescence. And this is calculated using a reference dye. So this is different to your dye, which is in your either probe or your cyber, which in my case, this is a probe, so this is FAM. We have a reference dye called ROX. And this is often in the master mixes. And this controls for pipetting error and the amount of effectively polymerase and stuff that you add into the sample, add into your conditions. And so what will happen, this is with the trace of this. And you can see here, there's a slight, very, very small difference in ROX, which then means that there's a small difference here. And obviously, PCR being exponential only takes a tiny, tiny amount to then throw. And so what then happens is this signal is normalized to this signal, and then you get the real signal here, which then shows the traces as they should be here. So just a heads up that you often see this is what's going on in the background, and this is what you'll see. So once we run our qPCR, we have our controls. What we need to do is make sure we QC the data properly. So we make sure we get the expected data and that we see the correct technical replicates. And so, for example, here we're having technical triplicates. So these are our 3 traces. And then what we should see is obviously when we overlay them, they all go on top of each other like this. And so this would be an example of good technical replicates. This is what we should see in every single one of our technical replicates, because ultimately it's exactly the same sample and material within them. So we should see that. What you shouldn't see, I'll show you later, is where it spreads. Negative controls, again, we should see a flat line. Sometimes you might see signal around like 40 or something like that. But what's important, if you do see signal very late, is that it's a lot later than your actual samples themselves, so that where you're expecting to see signal. If there's at least sort of 5 cycles between them, then you're pretty happy that what you're seeing in your samples is real. However, if you get a comparable signal in your negative controls, then that would suggest you contaminated some of your reagents. So go back, clean everything, start again. In addition to negative controls, it's also really important to not detect false positives, or to make sure that you trust all your data. And so, for example, an example of this is late amplification. So in general, we don't really trust data over 35 cycles. Now, obviously, if you use the right negative controls and they don't come up and you're seeing consistent signal at 38, then you can be more confident about that. But that's an example of where the controls are so important. So you can use those negative controls to assess if your signal is real. You get variation within technical replicates. So you can see here, for example, these are 3 technical replicates that have all come up at vastly different. So 1 hasn't even amplified, and 2 here are around a cycle, 2 cycles difference. That's up between 2 and 4-fold difference. However, the important thing here is you need to eyeball your data, because in this example, this would give me data. It would give me a CT, which I could then put into my downstream analysis. But what's really crucial here is obviously this isn't reliable. You really need to make sure that you QC your data, because you cannot use this going forward. if you take home one thing, it's QC your data and make sure your curves are on top of each other, and make sure that you do not use data which looks like this. So in general, there's sort of identification for false positives. There's a very rarely a reason to change the threshold yourself. Don't do it unless you have a really, really good reason to. There's a very famous example of where this has been done to give false positives. I'm not going to go into more about that, but I'm sure many of you know about it. And also, negative controls are really important to look at contaminants, because obviously contaminants can skew your data. So what I've got here is the list of the MICI guidelines. And so these are the minimum information for publication of quantitative real-time PCR experiments. You can see more about it in this publication here, but effectively you should be able to explain every single one of these steps in your methods paper, in your methods section of your paper. And if you can do that, then it means you've set up a good PCR. So what I thought I'd do to finish my section of this is to give a worked example of sort of how would we look at how does a gene of interest change upon treatment, and then how do the maths work after it, and then Chris will obviously take this forward in much more detail. So for example, we've taken our human cells, we've treated them with a drug which we think might be suitable for disease, and then we've extracted RNA using our optimized protocol. We've then performed the reverse transcription reaction and then qPCR using our, in this case, a probe-based chemistry. And so the first thing we want to do is I mentioned reference genes. So reference genes are what we then use to calculate relative expression. So qPCR, unless you can do it so you can get sort of copy number, but obviously in general it's used for relative expression. And this is the expression of your gene of interest relative to your control. And that's why it's so important that your reference gene, or your control gene, or it's also referred to as a housekeeping gene, is consistent and is stable between your conditions. So in general, in our trace, again, we see, obviously, we're seeing our trace. If it peaks towards the left-hand side, we have higher expression because it takes less cycles to amplify enough PCR product to then give a big enough fluorescence, which means we're starting with more material because we need less cycles to amplify it. Whereas if it's up here, we have less copies in the starting material, we need to amplify more to get it. Okay, so the first thing we do is we use the machine. The machine set the threshold. We have our replicates, which look really nice and on top of each other, and the machine will tell us what our control CT is, so that the number of cycles at which it crosses the threshold. So here for our control, we have 17.024. We've also then done this on our treatment condition. Traces look good, and this is telling us 17.078. So this is really good. This is really nice and consistent, and this is what we should be expecting to see, given that we put the same amount of material into the qPCR. Our next step is to obviously then look at our gene of interest, which may or may not vary, okay, upon treatment. And so what we have here is we have exactly the same. You can see that these are appearing further towards the right-hand side, which means they're at lower level expression than our reference gene, which is a very standard thing. And you can see here that our gene of interest in our control conditions is 29.8, so still well within the realms of suitable, good technical replicates. And then our gene of interest in our treatment. So this is the gene that we think might be changing upon our treatment. And again, technical replicates are fine. And what we see here is 22.5. So we see that this has shifted towards the left-hand side. So the cycle threshold has decreased, which actually gives us an idea that there is then more of that gene in our treatment than our control. But what we need to then do is calculate how much more. And so to do that, we use a process known as the delta-delta-CT method. And so the first step is the delta CT. So this represents the change in CT. And this is calculated by our gene of interest CT minus our reference CT here. So effectively what we're asking is how much does our gene of interest, the CT of our gene of interest, so here, for example, differ from our reference in our treatment condition compared to in our control condition. So this is what we're subtracting here, okay, here to here. And so then under normal conditions, we'd expect there to be 12.7 cycles between our gene of interest and our reference. However, under our treatment, we're suddenly seeing this drop. So we're seeing 5.4 cycle difference, right? Which would, again, is suggesting to us that there is more starting material because it takes less cycles to amplify it. How does that change in CT differ between our control and our treatment? And that's a step known as the delta-delta-CT. And so that's the delta-CT of your sample minus the delta-CT of your control. Well, so by sample here, I mean treatment condition, minus your control delta-CT. Okay, so what we do here, we subtract, in this case with a single replicate, we subtract delta-CT from itself, which gives us 0. but then we also subtract our treatment delta CT from our control, and here we get a change of 7.3 cycles. Now, when you have multiple replicates, what you'd be using is the individual either control or treatment CT, delta CT. What you should then subtract the average of your control group. So how many replicates you have? So you have 6 replicates, you take the average of those 6 replicates. And so Chris is going to go through this in more detail. It should become a bit clearer later, but this is just a general overview of how the analysis runs. So we have our delta CT, which is great. So we're seeing that it takes 7 less cycles to amplify enough, which suggests that there's a lot more. So what we can then do is calculate the fold difference. And so finally, to calculate the fold difference, because qPCR is exponential, we do a 2 to the power of our minus delta CT. And in this case, we're effectively setting the fold difference of the control to 1. What we see is 7, basically 2 to the 7.3. We end up with a fold difference of 164. So that means there's 164 times more transcript of this gene of interest in our treatment versus control. So as an overview, this is our sort of the analysis summary. So we identify the CT of interest of our reference and our gene of interest. We then calculate the delta CT by subtracting the reference gene from the gene of interest. We then calculate the delta CT by subtracting the average control from delta CT from the sample itself, the delta CT of that, and then we calculate the fold difference by using 2 to the power of minus delta CT in this method. So you might also see you do it the other way around. So here we've subtracted the reference gene from the gene of interest. However, you can also do it the other way around, where you subtract the gene of interest from the reference, but the important thing here is that instead of 2 to the minus, you just do 2 to the power of delta CT. So it doesn't matter which way you do it, you just have to make sure you get this calculation the right way around. And obviously, a good way to check that is by looking at your numbers. If your cycle threshold, if your delta CT is getting smaller, then your fold difference should be greater. And this is a good way to just check you've done this the right way around. Because if you do it the wrong way around, it'll look the other way. It'll go the other way around. So I hope that's been helpful in terms of setting up how to optimize and plan and perform a good qPCR experiment. And then Chris is going to take this analysis and sort of the data presentation a lot further forward and give you some worked examples as well later on. So if anyone has any questions, then I'm happy to take them. If you don't have any questions or you think of some later, then I'm happy to receive any emails about it as well.
Okay, thank you very much. So I think we've had a few questions pop up in the chat during the session and then another couple just now. So Ben, could you go through those for us?
Yeah, no problem. So the first one was our COVID PCR test based on probe or cyber. So I'm not 100% sure on this. As far as I remember, I'm pretty sure the main one is based on a probe based approach, but I think people would have designed tests for both cyber and probe. But yeah, I've never done it myself, and I'm not entirely sure. But I think the first one might have been probe based. And the second question being, how good is the reverse transcription of degraded mRNA from archived tumors? So obviously, degraded RNA is a big problem with qPCR. So in terms of reverse transcription, obviously an oligo-DT based won't work because that will only reverse transcribe intact RNA. So you'd have to use random primers or random hexamer approach. The big problem with degraded RNA is obviously you're relying on the amplicon still being present. And so that's going to vary massively dependent on how degraded your RNA is. So you'd have to test that out and perform probably serodilutions and all sorts of things to make sure that you're confident that what you're picking up is representative of the original sample. But yeah, degraded RNA is a nightmare. And I know from archived tumors and samples, we've had the same problem in some of our stuff before as well. There was one about the CT depends on. So yeah, yes, pretty much. So yeah, the threshold is set at the point at which the fluorescence itself goes above a background, and then at the time at which that detection becomes exponential, because then it's telling you that what you're detecting is the exponential nature of the PCR. And the threshold is set at the point at which the machine determines that those sort of parameters are being met. And then got one from Fabio. So the threshold is, again, because it's above a background, that's going to vary experiment to experiment depending on your pipetting or whatever during every experiment that you perform. So you don't need to make sure your threshold is exactly the same. It should be consistent, obviously, between it. There might be some decimal points in each probe, or depending on how you do it, each primer pair will have its own threshold, depending on the efficiency of that as well. But yeah, so across plates, there's no problem with doing it across different plates. But yeah, so then the threshold is determined for that specific reaction. Like I say, it should be relatively consistent, but there shouldn't be too much of a problem there. Thank you very much, Chris Donaldson, for saying it is probe-based. So in terms of how do you deal with variability in the control, what do you mean so whoever sent that in, do you mean in terms of your actual experimental controls, or your negative controls, or your, I assume it's experimental controls? Obviously natural variation, sorry Alison, you got?
Yeah, I mean, I haven't got any more information on that, but my take on it was almost sort of variability of the sample. So if you know, if you've got a different swab from the same individual and it came out with a slightly different, yeah. So how would you deal with that kind of variability? Yes. Okay.
Yeah. Okay. Yeah. So I mean, obviously, I don't know what you use to work on them. And obviously, humans are incredibly variable, and so that's when what Chris will talk to you about later, I suppose, in terms of the statistics will be very important there. Obviously, if you're working on samples which are very consistent, it's much more simple. But I mean, I wouldn't expect multiple biological replicates to have exactly the same CT. I'd then question that data enormously. So see, you have to set your experiment up so that you're able to determine a difference between your control and your test samples based on the variation that you're going to get. And as I say, Chris will talk about that later on. So I hope that helps.
I think we have one more question from Jeff. Have you seen that one, Ben?
Oh yeah, sorry. So if you're looking, so that would be similar to if you're trying to do copy number, I'd imagine. So you'd need a sort of a standard curve to then read off against. That's what we've done with trying to work out copy numbers. We put known amounts of material in and see what CT that responds to. So if you have a known amount of virus, for example, if you then do a serial dilution across a very large scale, then you can extrapolate the CT. You have your standard curve, and then you can extrapolate standard curve based off that using the CTs. And that's how we work for, as I say, for estimating sort of actual amounts of copy numbers and stuff in the cytoplasm. Yeah.
Okay, so I think we've gone through everything in the chat as it stands. So thank you, Ben. That was really, really good, really interesting. Chris, if you want to carry on with the session for us.
Okay, so I'm going to be talking about the design and analysis of qPCR experiments from a statistical perspective. My talk is broken down into 5 sections, and I'm going to go through these in the order that you should go through these things for a real experiment. So firstly, we're going to look at determining what the aim of the experiment is and defining the hypotheses that we want to test. Then we'll look at determining the appropriate statistical analysis to test these hypotheses. And then the next thing we'll look at, we'll be using the first 2 things to determine how many samples we'll need for the experiment. Then after data collection, we'll look at performing the analysis and interpreting the results. And then finally, we'll look at presenting the qPCR results graphically. So firstly, we'll look at determining the aim of the experiment and defining the hypotheses. qPCR is good for comparing gene expression between groups. So a typical research question for a qPCR experiment would be something like, is gene A expressed differently in conditions 1 and 2? Conditions 1 and 2 can be made up of independent groups where you have 2 groups of individuals you want to compare to each other. For example, in a experiment where you have a group of controls and a group with a certain disease, and you want to compare the gene expression between the two. Or they could be in a repeated measures design where you have paired sample, where you have one group of individuals or samples, and you're measuring them at 2 time points, perhaps before or after an individual. And each individual serves as its own control. So you're comparing, say, time point 1 to the earlier time point. For every gene that you're interested in a pair of conditions, there's a null hypothesis, and that is the expression of the gene is the same in each condition. So our aim for doing the experiment is to gather enough data to challenge this hypothesis and either accept it or reject it and accept the alternative hypothesis, that is that the expression of the gene is different in each condition. So in the situations I mentioned previously, the null hypothesis is slightly different in each. For the independent groups example, the null hypothesis is the expression of gene A is the same in both groups. So that's the same as saying the mean in group 1 is the same as the mean in group 2. For the paired samples example, the null hypothesis is that there is no change in expression of gene A within individuals by time point 1. So the mean difference between time point 0 and time point 1 is 0. So we've determined the aim of the experiment will be to compare gene expression between groups using qPCR. Now we've defined null hypotheses for a couple of situations. So the next step is to determine the appropriate statistical analysis to do to test these null hypotheses and what data to do the testing on. So just a quick reminder, what we get out of qPCR is the delta-delta CTs, and these are relative normalized expression differences between control and treatment groups. These are measured in cycles or doublings. So a delta-delta CT of 1 is a difference of 1 cycle, and that means double the amount of product in one group compared to the other. A difference of 2 means there's 4 times as much product. And the relationship between the delta-delta CT and the amount of product is 2 to the power of delta-delta CT. So for example, if you have a delta-delta CT of 7, then that is 2 to the power of 7, or 128 times as much product in one group compared to the other. It's common to see delta-delta CTs converted into ratios or percentages when they're presented. So you might see a graph like this one on the right, where you have a control group and a treatment group. The level in the treatment group is 300% or 3 times higher than in the control group. And you might see another bar say treatment 2 here, which is 33% the level in the control group. But the issue with converting to ratios or percentages is that you get this effect where things that go down, the difference of 3 times here looks a lot smaller than when things go up by the equivalent amount. So although ratios and percentages can be intuitive enough when things go in one direction, they are awkward when things go in 2 directions. And the reason for this is that ratios where things go down, i.e. ratios below 1, get squashed into the area between 0 and 1, whereas ratios where things go up spread out anywhere between 1 and infinity. So the solution to this with ratios is to calculate the log of the ratio, or the most convenient is the log 2 of the ratio, because this spreads things out evenly either side of 0, which is equivalent to a ratio of 1. Conveniently for us, delta-delta CT is the log 2 ratio. So delta-delta CT is already on the scale that we want it on to do the analysis, unlike ratios and percentages. So we want to do the analysis on delta-delta CTs, but you don't have to interpret changes or differences with just delta-delta CTs. You can interpret them however you like. You could convert them back to ratios and have them spread out evenly. Or my preferred method is to think of things in fold difference, because this makes it easy to compare something that goes up, say, by 4-fold to something that goes down by an equivalent amount, which is not immediately apparent necessarily with ratios, but if you convert to fold differences, then it's quite straightforward to see equivalent increases and decreases. So when it comes to comparing the differences statistically, t-tests and ANOVAs are what's commonly used to compare expression levels between groups, and these are the appropriate choice when applied to delta-delta CT values. And the reason for when they're applied to delta-delta CT values is that t-tests and ANOVAs are parametric methods, and this means they make certain important assumptions about your data. These assumptions are that your data in each group are approximately normally distributed, and that the variance in each group is similar. And if these assumptions aren't met, then the results will not be a valid test of the null hypothesis. So just go to these assumptions in a little bit more detail. First is the data in each group are approximately normally distributed. So what you want to see is a distribution like this with equal spread either side of a central value, which you'll tend to see for delta delta CT spread around 0. You won't tend to see for a ratio because of the problem with things being squashed between 0 and 1 spread out between 1 and infinity, because with a ratio you often end up with a right skew distribution. And the other assumption is that the variances between groups are approximately equal. There's no hard rule for what approximately equal means. It's often taken to mean that one is not 3 times larger as the other. And this is much more likely to be the case when you have a normal distribution like this than if you have a right skew distribution like this. Often you won't actually collect enough data to test these assumptions directly, but it's still reasonable to base your assumptions on how you expect delta delta CTs to behave. And just a quick note that you can do statistical tests to test for normality and equal variances, but it's not really worth doing these tests and they can be misleading. The reason for this is statistical tests that compare distributions tend to be underpowered when you have a small amount of data and overpowered when you have a lot. But the assumptions are not that your data is normally distributed and the variances are equal, it's that it's approximately normally distributed and the variances are approximately equal. And it's enough to look at the data or think about how you think the delta delta CTs are going to behave to make that assumption. Because if you do a test on data that you've collected, depending on the amount of data you'll either get a low p-value or a high p-value that can be slightly misleading. So it's not worth doing these tests. So now we know what the null hypotheses are and that we're going to use t-tests or ANOVAs to test these hypotheses. We can use this information to determine how many samples we actually need for an experiment. So this is quite important, but unfortunately it's very uncommon to see sample size calculations for qPCR in lab papers. I'm just going to go through why this is a problem. Without a sample size calculation, we have no way of knowing how small a difference between groups could be detected, so we don't know the power of the statistical test. What that means is that biologically interesting differences could be missed, so we could get false negative results. And also, if the number of samples to collect is not defined ahead of time, then this allows decisions to be made while data collection is ongoing, and this can lead to bias because people might seek statistically significant results. And this can lead to non-existent differences being found or false positive results. So one concept we have to think about for this is power. So the power of a statistical test is the probability that the test will correctly reject the null hypothesis when there is a difference between groups. So in general, the more samples you have, the more power you have. But the smaller a difference we look for between the groups, the more samples we need to achieve an acceptable level of power. And the usual compromise between sample size required and the risk of incorrectly not rejecting the null hypothesis is to aim for about 80 to 90% power for an experiment. If we don't have enough power, we run the risk of getting false negative results, also known as type 2 error. To give an example of this, in the situation we're testing this null hypothesis that for a certain gene, the mean of the gene is the same control group and the treatment group. We go away and collect 3 samples for each group, do a t-test, compare the groups, we get a reasonably high p-value, which is insufficient evidence to reject the null hypothesis. But had we collected more data for each group, for example, 8 samples for each, we get a much smaller p-value for the same test, which is strong evidence against the null hypothesis. And in this situation, the real difference is twofold because these data are drawn from 2 different distributions with a twofold difference. So in this situation, on the left, we would have made a type 2 error because we would have not rejected the null hypothesis when there is a real difference. But it's worth noting that adding more samples and retesting is not actually a solution here, because if we do this, we actually run the risk of getting a type 1 error, which is a false positive result. The reason for this is that, for example, if I collect 3 samples for 2 conditions, such as on the last slide, perform a t-test to see if they're different, and say the p-value comes out as 0.11, I might think, oh, that's reasonably low. Maybe I should collect some more samples and see what happens. So I collect another couple of samples for each condition, perform the same test again, p-value is now 0.07. Oh, this is going in the right direction. So I collect 2 more samples and do the test again. So I do that, and now I have a p-value of 0.04. I think, oh, that's low enough for me to reject the null hypothesis. So at this point, I stop. But perhaps had I gone on to collect another 3 samples for each condition and perform the test again, maybe now the p-value would actually be much higher, and there would be no no difference. The problem here is that I'm at risk of making a type 1 error, of incorrectly rejecting the null hypothesis when there is no difference, for two reasons. The first is making decisions on data collection based on the p-value. So I've been going through looking at the p-value and thinking, oh, that's going down, I should collect more data. And the second is multiple testing, because every test you do, there's a certain chance that you're going to get a low p-value just by chance. And if that happens, like it happened here, so I got a low p-value, so I stopped, then that will lead me to incorrectly rejecting the null hypothesis when there is actually no real difference. So how can we minimize the risk of these two errors? The answer to this is to perform a sample size calculation before data collection. This ensures any decisions on sample collection are not biased by the results you're getting. It also reduces the risk that you don't collect enough samples to detect the difference that you're interested in. And a third thing it does is reduces the risk that you waste resources by processing more samples than you actually need to. It's unfortunate that a lot of, as statisticians, something we hear quite a lot is people that come to us after they've actually collected their data. It's a slightly awkward situation, but you can still do analysis in this scenario, but you've got to be very careful about how you interpret the results, because you have to acknowledge that you don't know the power of the test that you've done. So you don't know if you're at risk of false negatives. And also you've got to acknowledge that if you make decisions on data collection based on the results, then you're also at risk of false positive results. Another question we often hear is, can I calculate the power of my experiment after doing analysis? The answer to this is yes, but it doesn't tell you any new information. If you calculate the power of a test after doing the test, it gives you no more information than the p-value already gives you. If you have a low p-value, your test will appear to have had high power, and if you have a high p-value, your test will appear to have low power. So what can you do? You can't do a lot for an experiment where you've already collected the data, but for the next experiment, you can do a sample size calculation and you can stick to it so you minimize the risk of causing either of these errors. So how do you actually do sample size calculations? So to do a sample size calculation, you need to make some decisions and you need to make some assumptions, and you need to estimate several parameters. The first thing you need to decide is whether you're doing independent groups or doing paired samples. This is the equivalent to deciding if you're doing a 2-sample t-test or a 1-sample t-test. You need to decide if your test is going to be 1-tailed or 2-tailed. For qPCR, usually that would be 2-tailed. Doing a 1-tailed test is equivalent to thinking that change can only go in one direction. And usually for gene expression analysis, that's quite a strong assumption, and it's not something you'd make. You also need to decide a significance level below which you'd think is the p-value is low enough to reject the null hypothesis. That's usually 0.05. You need to decide what power you want. Usually you'd aim between 80 and 90%. And you need to decide the allocation ratio between groups. Usually this would be 1, unless you might know that you're going to get more samples from one group than another. The final thing you need to estimate, which is usually the thing that people have most trouble with, is the effect size. This is calculated from the difference between the groups that you're looking for and also the variability within the groups. So the effect size we think about for sample size calculation is called Cohen's d. And basically the calculation comes down to the difference between the group means and the amount of variation within groups. So the larger difference between the means, the larger the effect size. And the smaller the variation within the groups, the larger the effect size. So this means that different combinations of variability within groups and differences between the groups can give the same effect size. So this is a graph, just an example of different ways you could end up with an effect size of 1. So it could be a relatively large difference between the groups with a large variability within the groups, such as the bar on the right here. Or it could be a relatively small difference between the groups with also a small variability within the groups, like this bar over here. And the same is true for any other effect size. So effect size of 2 here. It just different values of the difference and the variability will give different effect sizes. So when it comes to performing sample size calculations in practice, they're pretty straightforward to do if you use a piece of software such as GPower, which is free. It's slightly complicated to get into, but it comes with a manual and it's not too difficult. You just have to select the right test that you're doing, tell it what you want, such as power, and then you input all the things that I mentioned on the previous slide. It's also got something to calculate the effect size for you. So you could just put in the expected means of each group and the expected variability of each group. And from that, it will calculate the effect size and put that into the sample size calculation for you. So you can play around with this. You can try different scenarios with different amounts of power, different effect sizes, and see what sample size it recommends. This is just a graph to show that different effect sizes can lead to very different sample size requirements. So the red line here is 80% power and the blue line is 90% power. And as you can see, as the effect size gets bigger, the number of samples required to reach this level at each of these levels of power reduces quite considerably. And for very small effect sizes, it can be very, very large. And this does show that you can get down to very small sample sizes required to detect very large effect sizes between groups. However, I would probably not recommend ever doing as little as 3 samples per group, because if one of the samples for some reason comes out as there's something wrong with it, gives you a number that's not right, this will have quite a large effect on your average overall because you've only got 3 samples. So even in cases where you have apparently very high power, it does make sense to do more samples per group. And I would probably recommend never doing below 6 samples per group. But it's worth noting that there's some other things you might need to consider when thinking about sample size. One of those is attrition. It may be that the case that some samples you collect will not produce usable results, or perhaps you're recruiting participants and not all of these will provide samples. So you might need to inflate your sample size a bit to account for this. For example, if you expect 50% attrition and you need 100 samples in each group, then you need to recruit 200 to each group to get to 100. But obviously, attrition is a very contents-dependent thing. It's more of a risk when you're recruiting people, whereas if you're working on cell lines, you have a sample that doesn't work, you could probably just get another one, just grow up more cells and redo that sample, so attrition might not be an issue there. The other thing you need to think about is multiple testing, because the more statistical tests you do, the greater the risk of false positives just by chance. So to reduce this risk, you can adjust your p-value based on the number of tests that you perform. But doing this, it reduces the risk of false positives, but in doing that, it increases the risk of false negatives because it reduces your power. So you may need to inflate sample size to ensure that you still meet an acceptable level of power. Balancing type 1 and type 2 errors is a very context-dependent thing, and it often comes down to how exploratory the study that you're doing is. You may be willing to tolerate the risk of false positives because you don't want to miss out on anything, for example. Now that we've determined how many samples we needed for the qPCR experiment, you can go away and do the hard part, do the lab work, and collect the data. Once we've got the data collected, we can do the analysis and interpret the results. So the analysis that's performed should be what the sample size calculation is based on, which will usually be a t-test for pairwise group comparisons, or might be ANOVAs if you've got a lot of groups. Actually, practically performing the test is not too complicated. I'm not going to go into too much detail with that because it's pretty straightforward, and these are standard tests that are available in any stats package. I'm going to concentrate more on the interpretation here. I'm going to start off by talking about p-values. The reason I'm starting with p-values is that they've been quite extensively misused over the years, and they've got quite a bad reputation. But when they're interpreted properly, they do convey useful information. So a p-value is the probability that you would observe the results that you did, or more extreme results, if the null hypothesis is true. So if you have a p-value of, say, 0.32, that literally means there's a 32% chance that you would observe these results, or more extreme results, if the null hypothesis is true. So this means that the smaller the p-value is, the more evidence you have against the null hypothesis. So interpreting p-values can give you a lot of information. You should not interpret p-values based on which side of 0.05 they are. Although a 5% significance level is common, it's completely arbitrary, and it's an unreasonable simplification. Because if you consider a p-value of 0.049 and a p-value of 0.051, they're just either side of 0.05. So it seems silly to label two such similar p-values as either significant or not significant. What you should do is consider the magnitude of the p-value rather than just which side of 0.05 it is. I tend to break it down into slightly more groups to sort of quantify the evidence, the strength of the evidence against the null hypothesis. So very low p-values will give you very strong evidence against a null hypothesis, and as the p-value gets higher, the evidence you have gets weaker until we get to the point where you have little or no evidence against a null hypothesis. And another thing to note is you should not interpret p-values as meaning the difference that you observe is important. You need to look at the size of the difference and interpret what this means biologically or clinically, because p-values tell you nothing about biological importance. And as we've previously mentioned, you should not make data collection decisions based on p-values or analysis method decisions based on p-values, because this will inevitably lead to bias. I spent a couple of slides talking about p-values, but they're only actually a small part of the statistical analysis. So I'm going to go through looking at the results that come out of a t-test and an ANOVA just to show you what areas to focus on. So this is what you'll be doing after you've got your data into a stats package. For example, GraphPad up here, where you'll have your data arranged in columns for each treatment group and control group. Or in pretty much any other stats package, you'll have it arranged with each sample having their own row, having a variable for delta delta CT and another variable that defines the group they're in. So I'm going to show results that come out of GraphPad, but you'll get the same results out of any stats package for running these tests. So here we're comparing control group to the treatment group 1 using an unpaired t-test. So we get quite a lot of results out, but the things that we really need to focus on particularly is how big the difference is between the groups. And it also gives us the p-value up here. So looking here, we can see that the mean of column B is 3.67 and the mean of column A is 0, which we would expect it to be for this kind of experiment. So what we're interested in is the difference between the means. And we've also got the 95% confidence interval here, which is important for assessing how precise your estimate of the difference between the means is. So here, if we wanted to write out these results, we could say that there's very strong evidence that the expression level of gene A is increased by 12.8-fold on average, with a 95% CI of 6.3 to 25.8 in the treatment group 1 compared to the control group. You can see that the analysis here is done on the delta delta CTs, but you don't have to interpret them directly as delta delta CTs. Here I've interpreted them as fold difference, which I think is a bit more intuitive. And then you get similar results for an ANOVA, but there's kind of 2 stages to an ANOVA, because you have the ability with an ANOVA to do multiple comparisons within the same model rather than just look at 2 groups at a time. GraphPad presents the results from each stage in separate tabs. The first stage, there's not a lot of this that's particularly useful information directly, but what you're looking at here is this p-value, because this is testing the hypothesis that all of the groups that you put into the ANOVA have the same mean. So a low p-value indicates that at least one of those groups has a different mean from the others, but this stage doesn't tell you which is different. So here, if you have a low p-value, it makes sense to do multiple comparisons to find out which group varies from which other groups. But if you have a high p-value, there's not a lot of point in doing multiple comparisons, because you already know that all of the groups you put in have the same mean. So the second tab up here is the multiple comparisons. There's a lot of different ways you can select what multiple comparisons you're doing. Here I've kept it quite simple, and we've got control versus treatment 1 and control versus treatment 2. So this is almost equivalent to doing 2 t-tests. But in situations where you want to do a lot of multiple comparisons, it's better to do it in an ANOVA. And the reason for this is here we've got the estimate of the mean difference again for each group compared to control, but we also get a 95% confidence interval and p-value, and the 95% confidence interval here and the p-value have been adjusted for the number of comparisons that you've made, and this is important to reduce the risk of type 1 error if you're doing a lot of comparisons. So, but it's worth bearing in mind that the more comparisons you do, the more power you lose. So here we can see that, similar to what we saw on the last slide, there is very strong evidence the expression of gene A is increased by 12.8-fold on average, with a slightly wider 95% confidence interval than before compared to the control group. And we can see that treatment group 2 here, there is little evidence for a difference in expression treatment group 2, as the difference is very small, it's just 1.2-fold, and the 95% confidence interval includes 1 compared to the control group. So the main important results you should take from any t-test or ANOVA are the size of the difference and 95% confidence interval, because this lets you know how good your estimate of this difference is. So we've collected and analysed the results. So the last thing we need to think about is presenting qPCR results graphically. Unfortunately, in a lot of papers, there are a lot of common errors you'll see in qPCR result graphs. So I'm going to show this graph, which has several errors in it, and then I'm going to work through solving these errors to show you how to present a good graph at the end. So there's 5 errors in this graph that you'll probably be able to spot some of them quite easily, but I'm going to go through what they are. The first is that it's on a percentage or ratio scale, which we saw earlier was a problem. We've got error bars on here, but they're only going in one direction. There's a control bar on here, which for some reason doesn't have an error bar. And then someone's tried to show comparisons between groups on here, but it's not clear what's being compared to what. And the final problem with this graph is that we're using bars. And this is very common, but if we think about it, there's actually not really any good reason to be using bars on graphs like this. So I'm going to go through these errors in order and just show how we can fix them to end up with a better graph at the end. The first 2 are quite simple. We can just change from using a linear scale with a ratio on it, and instead we can plot the delta-delta CTs directly. And then we can interpret those how we want. So I would suggest interpret those as full difference. And we can also solve the error bar problem simply by just setting the software to give us error bars in both directions. So the next problem is the control bar with no error bar, and this is something that can crop up for a couple of reasons depending on the design of the experiment. If the analysis is unpaired, so if you have independent groups, then this control bar should have an error bar, and it could be that there's a problem with the way that the delta-delta CTs for the controlled group have been calculated. Whereas if this is a paired analysis, this bar is actually unnecessary because if you have paired samples, effectively what you're actually doing, the one sample t-test and comparing the treatment group to a set value of 1, and there's no error around that value. So in this situation, we don't actually need to show the error bar. We can just show, because we know the line at 1 is what we're comparing to, so we don't need a bar for that. The next point was the ambiguous group comparisons. So simple comparisons could be shown on graphs, but you've got to do it clearly, so it's obvious what's being compared to what. Here we have 2 comparisons back to the control. But you don't necessarily need to actually show these on the graph if they get too complicated. You could consider showing them in a table instead. And here you've got the ability to add a bit more data as well, not just say the p-value or some stars. You can show what the mean fold difference is. You can show the 95% confidence interval and number of samples in each group, as well as the p-value. And you can do that for each comparison in a more sort of concise and convenient fashion. And the final point was why are we using bars for these graphs? There's actually better ways to display this data that gives us more information visually. So all the information we get from a graph with bars on it is the mean, and the bar stops at the mean. But it doesn't tell, we can't immediately see from a graph like this how many samples are in each group or anything like that, or whether samples fall, or whether a mean is a good summary of that group. So instead we could use something like box and whisker plots, and these give a lot better idea of the distribution of points. And you can show things like the mean on here. So we've got the median as the solid line and the mean as the plus symbol. Or even better, we could just show every point. This is particularly good if we don't have a lot of points, which is often the case. So here we have the mean as the line, and we can see every point for each group. So we can immediately see this group has more samples in it than this group. And then graphs like that can be very easily extended to see the results from many more genes and multiple conditions, and they can be sort of presented in quite concise formats with both genes that go up by a considerable amount and down by a considerable amount on the same graph. So I mentioned error bars for graphs, but I haven't talked much about what to actually show with the error bars. You can show different things with the error bars, and you can show whatever you want really, but it's important to make sure you state what you show and that you're consistent between graphs. Otherwise, you can end up with something that's quite misleading. You've got to bear in mind that the choice of error bars greatly affect the appearance of the graph. And often the best choice is one that conveys useful information. So it's worth not just picking something that somebody else has used before, but think about what you actually want to use as the error bar. And I'm going to go through some examples of what you can use. So a quite common one to see is the standard deviation. This is a summary of variation from the mean, and it's good for visualizing that. Usually when you use a standard deviation, you end up with quite wide error bars, but it's good for showing the variability of each of your samples. The next thing that's often used is standard error of the mean. I think, unfortunately, this tends to often be used because it gives quite small error bars rather than to show what it's actually good for showing. So the standard error of the mean gives you an idea of how precise your estimate of the population mean is, and this is good and it's good at visualizing that. But using them because they give a tighter error bar is not a good reason for using standard error. My preference is to use the 95% confidence interval as the error bar, because informally speaking, the 95% confidence interval represents a range within which the true value of the population mean lies. So this is useful for immediately pairing samples or comparing groups to other groups. Because if you have 95% confidence intervals that do not overlap, you know that the p-value of a test comparing the means for those two groups will be less than 0.05. Whereas if they do overlap, you know it'll be over 0.05. And it also gives you a good idea of how precise your estimate of the mean is. But you don't necessarily need to even show those types of error bars. If you use box and whisker plots, the limits of these, they don't have error bars exactly. They just show the percentiles of the distribution. It can be, you can set what these are. Often they'll be between the 10th and 90th for the thin line here. And they'll show the 25th, 75th as the extremes of the box. They will show the median as the 50th percentile. And you can also add the mean on as well, just to show that the mean is near the median. And these are good for showing the overall distribution of groups, particularly when you have a lot of samples. Or you could use something like a kernel density plot, which is quite similar. It just varies the thickness of the box to show you where the points are located. So where we've got a thin part here, there aren't many points in this area, but where we've got a wide part over here, there's a lot of points. This is good for visualizing where your sample measurements fall on the scale. And it again is good when you have a lot of points. And then you could even use something like a heat map if you're doing enough genes and enough treatments or enough conditions to make this a feasible option. You often see these for things like for large throughput experiments like sequencing experiments, but you can use them for qPCR on a smaller scale if you want to. And these are good to use alongside techniques like hierarchical clustering, where you're seeing which samples group together. I'll just quickly talk about showing p-values on graphs. In lab papers, it's common to see stars used to do this, but there's no standard meaning for these stars. So if you do use them, you have to make sure they're clearly defined in the figure legend. Although these do convey p-value magnitudes to some extent, only usually up to 0.05, and then usually anything above that is considered not significant and either not labeled or just labeled as NS. And they can get a bit complicated when you show a lot of comparisons on a graph at the same time. So I think a useful alternative to this is to show the 95% confidence intervals instead. And then rather than having the stars or comparisons shown directly on the graph, you can show a summary of each comparison and its p-value and other summary statistics in a separate table. And it's always worth remembering the graphs are overall visual summaries, and you don't have to show p-values on the graph because that's only part of the analysis and it's not a particularly visual one. So all the graphs that I've shown in this presentation have come from GraphPad, and I think that's probably the software that I'd recommend, because you can do the analysis in the software and then you can produce publication quality graphs directly from GraphPad. And also it's quite a common piece of software that lab people are familiar with. But it can also be useful to have a vector graphics editor to put together figures. So if you want to put together multiple graphs into a figure, it's quite useful to have another program to do this. And I'd recommend Inkscape or Adobe Illustrator. So I've been through all these sort of statistical parts of a qPCR experiment right from the start, determining the hypotheses, figuring out how many samples you need, performing the analysis, and presenting the results. So just to summarize, I would say it's always worthwhile planning ahead. You need to consider what analysis you're going to do before you start, so you can have a sample size calculation and know you've collected enough data, or know you're going to collect enough data to do a credible experiment. And right at the start is the time when a statistician can be helpful to you, rather than later on when you've already collected the data. They can particularly help with doing sample size calculations, because that can be one of the bigger hurdles to get started. You should always perform the analysis on the delta-delta CT values and not on ratios or percentage or anything like that, because the delta-delta CT values will have appropriate distributions, whereas percentages and ratios won't. And always interpret results in the biological context and think about how big the difference between the groups is and what it means for whatever area you're working in. And don't just think about things as statistically significant or not significant. And it's best to try and avoid this kind of language entirely. It's still fine to use p-values, but think of them as a measure of evidence against a null hypothesis, and don't just dichotomize them. And when it comes to presenting graphs, present the delta-delta CT values on graphs, interpret them as the full difference. It's best to show all points if you can, and show the 95% confidence intervals around the mean. And just give a couple of sources for further information. There's a couple of previous outputs from the NHR lab stats group that would be useful, not just for qPCR, but for any kind of lab-based research. There's 2 papers in eLife that are worth looking at about units of analysis, and also RIPOST, which is a framework for improving the design and reproducibility of lab-based research. And also recommend having a look at the GraphPad Prism user guide, because it's very user-friendly. The software is made by lab people, so they understand what kind of experiments are being done in labs, so they explain statistics in enough detail to be useful, but not in overwhelming detail, and they do it with an understanding of the lab perspective.
Okay, thanks, Chris. So we do have it scheduled that we'll go straight into an example, and I know that there are some questions coming in, so I think it's your choice as to whether you want to tackle a few of those now. We are a couple of minutes sort of early running.
I think it's probably best if we do the example bit now and then do the questions at the end, I think.
Great.
Yeah, so the next bit is a kind of a very quick practical example of going from doing the qPCR to getting the graph at the end, going through the analysis and actually practically calculating delta-delta CTs. I'll share that now.
Okay, but please do keep your questions coming in, and we'll have time to look at those at the end.
I'm going to quickly run through an example analysis of a qPCR experiment, as there are some important technical points that can only really be explained with example data. So I'm going to cover 4 main points. The first is going to be combining multiple qPCR runs, then practical calculation of delta-delta CTs in Excel, followed by analysis of these delta-delta CTs in GraphPad Prism using a one-way ANOVA, and finally creating graphs in GraphPad Prism and combining them into a figure in Inkscape. It's common to have to combine results from multiple qPCR runs, as you often cannot fit all of your samples and genes of interest into one run. Some qPCR software lets you combine multiple runs for analysis, but I'm going to show you how to do this manually using Excel. In this example experiment, we're going to have 3 conditions. There's going to be 1 control group and 2 treatment groups, A and B. There are 3 genes. One is the reference gene or normalizer, and 2 are genes of interest called X and Y. There's 6 biological replicates for each group and gene combination, and for each of these biological replicates there's 4 technical replicates. So the CT we're going to use for each biological replicate will be the mean CT of the 4 technical replicates. And this data was collected in 3 separate runs. So here we have the 3 run files for our experiment. I'm going to open up run 1. So run 1 contains data for the reference gene, gene X and gene Y for 3 of the control group replicates and 3 of the treatment A biological replicates. So the other 3 replicates for the control group, the other 3 replicates for the treatment group A, and the 6 replicates for treatment group B are in the other 2 files. So the software gives us a lot of data about the run, but all we're really interested in is the CT value for each biological replicate. So for example, these 4 technical replicates make up the reference gene for biological replicate number 2, and we're going to be taking the value of where the average of these lines crosses the threshold. So we can export a lot of data from this software, but all we actually need is the sample name, target name, and the mean CT. I'm just going to copy this data into Excel. And I'm going to copy the CTs over into this template that I've made, which has a sheet for gene X and a sheet for gene Y. And then for the control group, we copy in the reference CT, gene X CTs. Treatment group A, we copy the reference CT, gene X CTs, and the same for treatment group B. And then we do the same for the gene Y CTs on the other sheet. So I'm just going to copy these values over. So I've copied over the reference CTs for treatment A and control and the gene X CTs. And I'm going to do the same for gene Y and the other replicates and for treatment B replicates from the other experiments. So now I have all the CTs entered into the sheet for gene X on here and for gene Y on the other page. So now we need to calculate the delta CTs and the delta delta CTs. So to calculate the delta CT, take the reference CT minus gene X CT. We do the same for each of the biological replicates. And we do the same for treatment A, treatment B. The next step is to calculate the delta delta CTs, and for these we need to have the average of the delta CT in the control group. And we calculate the delta delta CT as the delta CT minus the delta CT average. We want to do the same for each of these, so I'm going to put a $sign here so it doesn't change when I auto-complete these cells. You can see for all of them it's this delta CT minus the delta CT average. Our average delta delta CT for the control group will be 0. So now we do the same for each of the treatment groups, except we take the delta CT for the treatment group minus the average delta CT for the control group. And now we have our delta delta CTs for each of the treatment groups and the control group. So I've also done the same for gene Y to calculate the delta delta CTs for each group. And now we're ready to put these into GraphPad to do some analysis and to make some graphs. In GraphPad, we're going to be using a column type sheet and we're going to be entering our replicate values into columns. So we need one sheet per gene and a column for each group. So I'm just going to copy over the delta delta CTs for gene X into GraphPad. And I'm going to create a new sheet to do the same with gene Y. So now we've got our delta delta CTs in GraphPad. We've got 2 sheets, one for each gene, and we've got 2 graphs set up as default. So before we sort the graphs out, we're just going to do the analysis for these genes. We want to make 2 comparisons here. We want to compare treatment A to control and treatment B to control. So I'm going to do this using a one-way ANOVA. Our data is unpaired, and we're going to assume normality and assume equal variances. So on the multiple comparisons tab, we decide what columns we're going to compare to what. Here we're just going to compare both the treatment groups back to the control group, which is column A. So if we do this for gene X, we can see on the first sheet that we have a very low p-value for the ANOVA summary, and this means that not all of our groups have the same mean. So if we look at the multiple comparisons tab, we can see that treatment A is different from control, but treatment B is not very different from control. I'll do the same for gene Y. So the results are pretty much the same for gene Y. Treatment B is not different from control. Treatment A is. But for gene Y, there's a much bigger difference. So the next thing to do is to prepare the graphs for gene X and gene Y. And there's quite a lot of things we want to change from this default graph. I want to make these two graphs easily comparable, so I'm going to check that they're on the same axis. And set the gene X axis to be the same as for gene Y. I'm just going to extend the gene Y axis a little bit below the line as well. We're going to start to set this graph up how I want to see it. So first we're going to set the tick interval back to 2. And remember, this is delta delta CT on this axis, but I'm going to change it to display full difference. So to do that, I need to manually change the points on the line from delta delta CT to full difference. So at y equals 0, that's equivalent to a full difference of 1. A delta delta CT of 2 is equivalent to a full difference of 4. And I'm going to put a guideline for each of these just to make it slightly easier to see across the graph. So the next thing we want to change is the appearance of the data on the graph. And instead of bars, I'm going to use a scatter dot plot showing the mean and the 95% confidence interval. And we can change each data set to look how we want. So I'm going to set the color of the control in each of the treatment groups. And I'm going to bring the error bar in front of the points so we can see it more easily. So we have a quite nice graph for gene X showing the difference compared to the control group, the two treatment groups. And we're going to do the same for gene Y. You can make most of these changes using the magic wand tool. So this tool lets you take a graph and make it look like another graph. It often doesn't quite get everything. You can see here it's changed the axes, but it hasn't changed the bars to a scatter dot plot, so we have to do that manually. And it also hasn't changed the colors of the data sets. Now we have our graphs set up how we want them. We could combine them in GraphPad into a figure, but I prefer to do it in another program like Inkscape because it offers a bit more flexibility. So we're going to export the gene X and the gene Y graphs and open them in Inkscape. So we're going to hit export up here, and we're going to export these as an EMF plus file, which is a vector file. Just have to select where we want to export them to. Okay. And I'm going to do the same for gene Y. And open Inkscape. So we can just drag and drop our EMF files into Inkscape. So The default background guide on Inkscape is an A4 sheet, and it is working to actual size when you're making figures, because it makes it slightly easier if you're preparing something for a publication to think about how the figure might fit on a page, because usually journals prefer a double width figure or a single width figure, and they can be any sort of depth. So I'm just going to do some very simple changes to these. Just going to align them. Add on a title. I will export this as a PNG image at 300 dpi. And here we have our final figure ready for publication.
Okay, thank you. So, Chris, if you want to start going through some of those questions that we've seen in the chat.
Yeah, so there's been a few questions about things like not knowing that delta delta CTs are normally distributed. So, for example, there's one: have past studies supported the normality assumption of delta delta CT values? It's not really that you'll look for a study that has published saying delta delta CTs are normally distributed. It's more a thing that comes from experience of measuring these things and having a look at the data that you collect, and also what we expect biologically. What seems like a reasonable expectation for the distribution of these things. And also it comes from what we know about ratios, because we know that ratios aren't normally distributed. They tend to be right skew because of the issue of ratios. The stuff that fits between 0 and 1 is a very small area, and then there's this very large area to the right of 1. And if we take the log 2 of a ratio, that's how we make ratios normally distributed, and that tends to work. And delta delta CT is a log 2 ratio. So for what we know about delta delta CTs, essentially there's not, a lot of the time it comes down to there's not any reason to believe that they're not normally distributed. Because obviously when a lot of the time when you're doing lab work, often you don't collect enough data to sort of directly assess whether something's normally distributed. Because to do that, the best way to do it is visually, and to do that you need sort of tens of measurements, and often you don't have that many. So with this sort of thing, when you have small samples, it's best to think about what you expect to see, and then you can still look and see where your small number of samples are, and just see if there's any kind of anything that gives you any reason to doubt that. But often it's difficult to make it. Typically, it's pretty difficult to make any judgment, particularly with a small number of samples. So it's kind of reasonable to assume that delta delta CTs will be normally distributed based on just what we know about them. And you can check it visually if you have enough samples. I think there's another couple of questions about whether you can do the stats on the 2 to the power of delta delta CT values, i.e. ratios or percentages. And also there's questions about non-parametric tests. The thing to do, if you, in any situation, not just qPCR, if you have a situation where you have something that's not normally distributed, you, instead of using a t-test or an ANOVA, you can use a different method, such as a Mann-Whitney test would be the non-parametric alternative to a t-test, and the Kruskal-Wallis would be alternative to an ANOVA. And these are non-parametric tests, so they don't make the assumptions that your data is normally distributed and that the variances are similar between groups. So that sounds like a useful get out of assumptions free card, but it does come with some limitations. Because you lose a bit of power using these tests compared to t-tests or ANOVAs, and also they're not quite as, the interpretation interpretation is not quite as simple as people assume. A lot of people think that these tests compare medians, but that's only true in a specific situation. Those tests compare medians of groups if the distribution in each group is the same, but that isn't necessarily the case. So it's better to think of them as comparing two distributions to each other, which is not quite as simple to interpret as, say, saying that a mean is the same in another group compared to another. So often you might see in the literature, people will do what people tend to do is, instead of doing analysis on delta CT values and assuming they're normally distributed, is they convert to ratios and percentages and then decide they're not normally distributed. Often this is based on normality tests, which are not a good idea to ever do. They're pretty misleading. A lot of papers, you'll see a sort of robotic thing where people have done the normality test and looked at the p-value and just seen which side of 0.05 it is, decide whether they do t-tests and ANOVAs or Kruskal-Wallis and Mann-Whitney. But the problem is then they inevitably end up doing both, because they get some that seem normally distributed and some that don't. And that's a very awkward situation, because then they're not presenting something consistent. And it's particularly true for graphs, because they'll still present the mean and 95% confidence interval on a graph where they've done a Kruskal-Wallis test. But really, the mean, if you have something that's not normally distributed, the mean isn't a good description of that distribution. The median is. So if you were going to use that, you'd want to plot median and interquartile range on that graph. But often because there's mix and matching between the two, they end up just plotting mean and 95% confidence interval, but then doesn't match with the analysis, and it gets very awkward. But all that can be avoided by doing the analysis only on the delta CT values, because you can assume they're normally distributed. So you can do one type of test and you can present mean and 95% confidence interval on the graph. But that's not, I mean, in other situations, in other kinds of tests and things like that, you may legitimately have a non-normal distribution, and then Mann-Whitney U tests and Kruskal-Wallis tests are valid ways of analyzing the data. You just need to make sure you interpret correctly. They're not quite as simple as interpreting t-tests and ANOVAs. And then there's some questions on essentially on the assumptions around the sample size calculations, sort of how, particularly one thing trouble people have is figuring out the variability of their samples. It's quite easy to think, I want to see a certain difference between my samples. Often biologists think twofold is the minimum difference I'm interested in. That's quite simple, but the other part you need for the effect size is the variability within your groups, and that can be slightly more tricky to estimate. So sources that you can sort of look at to refine your estimates for that are previous experiments. Maybe you've done this on a small scale with the same genes that you're interested in. That can give you an idea of the variability, but you've got to remember that a small sample, you won't have a very precise estimate of variability. So don't take it as being the right answer. You want to be a bit more conservative. You could also look in the literature and see if people have done the same genes in different samples, maybe in cell lines or something else, and see what the level of variability is in that. One tricky thing with looking in the literature is people often don't present the data that you need to do that, which can be a bit inconvenient. But ultimately, the decision you come to for the variability, and anything you put in the sample size calculation is an assumption. So a sample size calculation is only ever a guide. And the better the data you can put in, the better the guide it is. But that's not to say that even if you have no data for your samples and that gene, there's probably something you can still think, well, I expect this level of variability in a biological system. And you might make that decision based on some sequencing data or just what you know about qPCR. But even if it's a weak assumption like that, the sample size calculation is still a useful guide, because it makes you think about the experiment all the way through, right at the start. So you've already decided what the analysis is going to be, and you'll have done your best to know that you're going to have sufficient power to detect differences that you're interested in. There's a question, does it make sense to use a t-test with any 3 samples per group? it's not possible to test assumptions such small samples. So yeah, that's another thing that comes down to knowing knowing how things are going to behave. But the n equals 3 per group is somewhat of a meme in lab sciences. I've had a look at a lot of qPCR papers, and often people will just say we did at least 3 biological replicates, or n equals 3, as if that's the right answer. But it's worth noting that doing a statistical test on 3 samples, even if it appears you have sufficient power, it's not very believable. Just because it's just such a small number of samples, you can't get a good estimate of the variability with just 3. And you've got the problem that I mentioned in the talk of that if any of those samples for some reason is incorrect, then it has quite an influence on the mean. So it can be, you might just end up getting a result that's different than it should be because one sample gave you a spurious result for some reason you didn't realize. So one point to emphasize is I don't think any statistician would ever say 3 is enough in a group. I think you always need to do more than that. There's a couple of comments on sample size being sensitive to the variance of the outcome and whether a small sample size can be representative of a very variable population. It's true. You can't, with a small sample size, you can't really get an estimate of the variability. And it's true that different genes in different situations will have different variabilities. So that's something you need to think about and something you need to take account of in sample size calculation. So often it means you have to think conservatively. And if you don't know what a variation, what variability to expect in a group, assume it's going to be reasonably large. So then you're more likely to end up having an overpowered test than an underpowered test that misses something. There's a comment, again, that's obviously easy to interpret, but doesn't that mean that any uncertainty in the mean of the control group is propagated the uncertainty in the estimates of delta CT for the treatment groups? So this comment suggests that one issue with qPCR is when you do a single run, often you can get, often the software will give you the expression levels of the genes compared to the reference gene in that run, but you don't have to do the reference genes or your test genes at the same time on a run. Usually you can't. And one I mentioned in the talk about one situation where you can have, sometimes you see a graph with a control group that doesn't have an error bar. And I think one way this comes about is that people take the wrong information from the software. Instead of taking the CTs and calculating delta CTs using the average of the control group, what they do is they take the control group from each run that they did, and then they take the delta CTs calculated per run. And what that does is effectively sets the delta CT of the control group to 0 with no variation. So in that case, the uncertainty control group is kind of moved into the treatment group. But the way around that is to make sure you calculate delta CTs based on the average delta CTs from the control group of all of your reference gene runs, not just one per experiment. There's one, do you usually run replicates of a single sample? If so, how do you decide if they are sufficiently reducible to be used? Ben touched on this in his talk. It's essentially, a bit of this comes down to experience. You have to know what you're looking for in qPCR. So if you have, I think Ben showed an example where there was four replicates, technical replicates that should give exactly the same result theoretically, but you get ones coming up at different times and a couple not coming up, then you're going to look at those and think the ones that are coming up on their own separately are kind of spurious. What you're looking for is consistency. So if you have, preferably as low cycle as possible, because lower cycles are more reliable, but you want, usually I'd say do a minimum of 3 technical replicates per biological replicate. I used to do 4, but you're looking for those technical replicates to be basically on top of each other. And if they're not, you may have an issue, and it may be that you're not measuring what you think you are, so you have to be careful. I don't know if you've got anything else to add to that, Ben.
No, all I was going to add is sort of in terms of an actual number. I mean, I would, if you're looking at technical replicates, realistically, you'd probably 0.25 of a CT, 0.5 absolute max. I'd be a bit worried if it was 0.5, but that if you want an actual number, that's what I would work off. Obviously, it all depends on what you're doing. But yeah, normally I'd want my technicals to be within 0.25 of a CT.
So in terms of estimating the expected effect size, how you sort of pilot study versus literature search, this is, as I said, well, this comes down to sort of, you have to use whatever data you have available. And if you don't have data available, look at other genes and other literature and come up with a conservative estimate to try and come up with some size calculation. In ANOVA analysis, there are different post-tests available. For example, it shows done, but there are others such as Bond-Ferroni. Can you explain differences in using different post-tests? Practically, the differences, I think the different post-tests that you have in GraphPad, I think practically it comes down to what groups you want to compare to what. I think it does different ones for if you want to do all comparisons, which probably you don't want to do, but may want to do in some cases. Or I think DUNS is the one where you select a control group and compare everything back to that. So I think the sort of the maths behind them, I don't know what the difference is top of my head, but it comes down to really what you want to do. So for post-test multiple comparisons, you want to do the minimum that answers your questions. So you don't want, if you're interested in comparing everything back to a control group, you don't necessarily need to do the comparisons between treatment groups because they might not add anything. So if you don't need to do those, don't do them. Just select the post-test that does the comparisons that you want to do. Some qPCR uses standard curves whilst others don't. Do you think there would be a difference in the variance within groups between the two approaches? When you do a standard curve, usually you're doing absolute quantification. So as Ben mentioned earlier, you're doing serial dilutions and you have a sample of known concentrations, of known copy number of something, like I don't know, a virus or something, or DNA. And then you're comparing your sample to that. So you do a standard curve of the known dilutions. So you have this like a line basically, and you see where your sample falls on that. So it gives you an absolute number that's in your sample. I think when it comes down to variability within the two approaches, because it's the same technology underneath, I think you'd probably be looking at similar levels of variability. The only thing is the absolute, you're talking in absolute copy number, whereas with relative quantification, you're talking relative. So because it's bulk qPCR, I think you're probably looking at about the same levels of variability with that. Sometimes labs give a level of quantification / detection figure. What does that mean? I'm not sure. That it could either be that they're saying that they're after a certain CT, they're not. They're saying anything that comes up after a certain CT is not detected, or it could be they're talking about where their threshold is set for determining what the CT is. But the second one I don't think would be particularly useful because usually that's determined per run, and you don't really want to change that. But do you know what they might be talking about if they show those figures, Ben?
No, I'm not familiar. As I say, yeah, unless it's detected below a certain, they might give a CT in terms of what they determine to be real, which is what I mentioned earlier, sort of, which is arbitrary. I mean, people tend to call 35, but obviously with the right negative controls and everything, you can push that. But yeah, I wouldn't know off the top of my head without actually seeing the figure themselves.
Another one, I may have missed you answer this, but in total, what is your recommendation for collecting samples of cells counting bacteria colonies before you do the analysis? So do you mean do you mean doing a kind of pilot to get an idea of the levels? Because if you're doing sort of absolute quantification, you kind of want to do the same thing. Is you want to have an idea of what level you'll be seeing the variability between different samples, so you can compare groups. It's just that in that case, you'll be thinking in terms of absolute number rather than a delta CT. So you could, instead of when you calculate the effect size, you'd be talking the difference will be an absolute number, like a million copies, 1,000 copies, or something like that, and standard deviation will be in number of copies as well. As a comment, I think for the previous question comment, I think we're talking about the limits of detection based on the fact it's hard to determine true zeros and limits of quantification when the level's below. Well, yeah, this is an issue with qPCR. If you run any qPCR for long enough, you will detect something, and different kits for different genes have different reliabilities. So some will be 4 and they'll come up, say, about 35 and give you a false positive, whereas others wouldn't come up until cycle 60 or something like that. So it's awkward when you want to say that something's not detected at all, because it's difficult to say that it isn't with qPCR just because of the fact that you keep amplifying. At some point, your probe's going to break down and you're going to get some sort of signal for some reason. So it is difficult to not detect something in qPCR. So it is tricky around the sort of, I guess people would often set a threshold that they say anything below, anything above cycle 35 often is just, we're going to say it's not detected if it comes up, because anything that does come up onto that will probably be very unreliable and spread out. Did anyone notice any other questions that I missed?
No, I haven't seen any others, and I think we are at time now. So I'd just like to thank Ben and Chris. I think it's been an excellent session today. So I'll give you the clap that comes up on Zoom there. So thank you very much. That has been absolutely excellent. Hopefully everyone's seen my message in the chat about giving feedback and how you can sign up for the mailing list. I'll just put that in there again if anyone wants to grab those links. But thank you so much to the speakers, and if anybody does want to get in touch, just send us an email and we can pass on any additional questions. Okay, thank you everyone. I'll see you soon.
Thank you.